Hi, everybody, and welcome to this week's lecture. And what we're going to be looking at and trying to understand today is going to be gene expression. Now, when it comes to gene expression, there's going to be two very important steps that we're going to be looking at. We're going to be looking at transcription and translation. So our learning objective is to just understand how cells make protein by transcription and translation. So before we can go ahead and get started, one of the very first things we need to understand is exactly what gene expression is. How are we going to go from DNA to proteins? What's going on? And exactly what steps are we going to be taking? So when it comes to gene expression, all we really need to know from this, or what this really means, is just how the cell is going to be using information in order to make proteins. Now, proteins are incredibly important, as you know, throughout this entire semester. We've been talking about them, and we understand that they're going to do very important jobs. And again, our body absolutely is going to need them. So we need a way to kind of make more whenever we have the need to. So when it comes to gene expression, again, it's going to occur in two steps. The first one is going to be transcription. Now, all transcription of essentially is going to be is just copying DNA information, and we're going to be using that or making something known as our messenger RNA. So as you know, our DNA is incredibly important as everything we have or everything we need in order to make certain things within the body. And we know that that's going to be found in the nucleus. But as you know, DNA is so important and so vital that we don't want it to leave the nucleus. We want it to stay exactly where it needs to be. So in order to take some of those instructions and send them to places so we know what kind of proteins we're producing, we're going to go ahead and make something known as our mRNA, messenger RNA. This will be able to leave the nucleus. Once that leaves the nucleus, we're going to go ahead and initiate our second step, which is going to be translation. Now, all translation is we're taking that mRNA message here, and we're going to translate that genetic information into a sequence of amino acids. Remember, our amino acids were our monomers, our smallest forms, for proteins. And again, we're going to take a bunch of our amino acids and chain them together to produce a polypeptide, or in this case, our protein. So this is what that's going to look like. You can see we have our DNA here. We have our specific gene that we're trying to go ahead and produce or express. And then we're going to go ahead and go through transcription. So we're going to go ahead and take our DNA, make a copy, which is going to be our mRNA. And then we're going to take our mRNA. We need to go ahead and translate this brand new code in order to make our proteins. So what's exactly going on? Where are they going to be located and where are they taking place? Now, when it comes to transcription, it's going to be solely regulated in the nucleus. The reason why is because DNA is going to be located inside the nucleus, and this is where transcription needs to happen. Remember, the DNA cannot leave the nucleus because, again, it's so important we want it to stay right in that center. But we're going to go ahead and create mRNA, which is going to be able to leave. So again, we're going to go ahead and make a copy kind of our DNA using a template or using it as a template, and then we're going to have a single strand that's going to be able to leave. So that's transcription. Now, translation is going to take place in the cytoplasm, and we're going to specifically be utilizing an organelle, ribosome, and this is where our proteins are going to be produced. This is where our mRNA is going to go ahead and slot in right here, and then we're going to have our tRNA and rRNA in order to chain together our amino acids in order to make our protein. All right, so let's go ahead and get started with our first step, which is going to be transcription. Now, let's go ahead and kind of see exactly what transcription is, and let's go and take a look at an analogy on how that would work. So let's say that we went ahead and had our DNA as a master book of instructions. So the analogy we're going to go ahead and use is going to be information or instructions for building and maintaining a city. Now, when it comes to DNA, DNA is going to be our copy or our master book of all the instructions. Now, there's only one copy of this master book, so we do not want to lose it. We want to make sure it's staying safe, and we're keeping it inside the nucleus. But we still need some of that information, and we need to make instructions so we can distribute it to workers for them to build and maintain the city. So what we're going to do is we're going to go and look through all of our instructions through our master book, and we're going to find the pieces that are going to be relevant. What we're going to do then is we're going to go ahead and make copies of those so then we can transport them and give them to those workers in order to produce and maintain our city. So it's going to be the same idea when it comes to DNA. Again, there's going to be specific parts within our book that we want to make copies of, and we want to give those instructions out. So now we have a bunch of these copies. which is fantastic. They can go ahead and use it and make those proteins, maintain the city, do all those things. Yet we still contain or still have retained our master book of instructions. So again, we have those safe whenever we need to reference them. But again, we're able to send those copies out. So that's kind of what transcription is going to be. So the next thing we need to identify when it comes to transcription is what exactly is RNA? What is it? What are we doing with it? And why is it different from DNA? So RNA is very similar to DNA, but there are going to be some important differences. One of the first one is RNA is going to be single-stranded versus DNA is double-stranded. So again, you can see right here, we have our RNA, our ribonucleic acid, and you can see it's going to be a single strand versus our DNA, which is going to be our deoxyribonucleic acid. Another major difference between the two is going to be the 4 bases and how they're going to pair together. Now, when it comes to DNA, remember, it's going to go A to T, so apples, trees, and C and G, cars and garages. Now, when it comes to RNA, we're still going to retain the C and G. The cars are still going to be found in garages, but the major difference is instead of thymine right here, we're going to be using something known as uracil, so something that's slightly different. So A is now going to bind with U. So this is going to be one of the major differences when it comes to our base pairing rule. Okay, so again, DNA, A goes with T, apples are found in trees, and then C and G, cars and garages. When it comes to RNA, instead of A and T, it's A and U. So again, that's our major difference when it comes to RNA for our base pair. So again, this is just another way of kind of seeing that over here. We have our DNA and RNA. So remember DNA, A, T, C, G. And then when it comes to RNA, A, U, C, and G. All right, so let's go ahead and take a look at the steps of transcription. So remember, when it comes to transcription, all we're trying to do is we're trying to make a copy of those instructions from the genes. Now, again, we do not want to lose our DNA. We don't want to mess with it, so we're going to go ahead and make a copy known as our mRNA. Remember, messenger RNA. And our messenger RNA is going to be used later in the cytoplasm to make those proteins. So what's going to happen when it comes to step 1 is we're going to have something known as an RNA polymerase. This is going to be kind of like a copy machine, and this is going to help facilitate the creation of our mRNA. So you can kind of see this right over here. Right there. Now, what's going to happen is our DNA is going to unzip and open up. Remember, those hydrogen bonds, which are just strong enough to keep everything together, but just weak enough to break, are going to open up. It's going to unzip. So let's say this is the part of the DNA we want to copy, and this is the part we're going to use as a template. Okay. One of the very cool things is because we have that specific base pairing rule, if we were to use this side or this side, we'd still be able to have the other side as well based upon the complementary piece. So again, the DNA double helix is going to open up so that the copying machine called our RNA polymerase can read and copy the DNA instructions found in the genes. So after step 1, in step 2, what's going to happen is one of the 2 strands of DNA is going to be used as our template, and the DNA instructions are copied using our base pairing rule of RNA to the DNA template. So remember, that's a very important thing because instead of thymine, we're using uracil. So this right here is our DNA template. And you can see that in this one, it's T, G, A, C, A, and T. Okay, so what's going to happen is we're going to start creating our mRNA. So our bases are going to come in. So T is going to bind with our A right here. So A and T go together. And then we have our G over here. So you can see we have our C that's going to come in. We have our A, and again, if this was DNA to DNA, this would have been a T. But because this is DNA to mRNA, it's going to be a U instead. And then our C goes with our G. And then you can see our U is coming in to bind or be our complementary piece to our A. So we have our U here. And then finally our T, which again would bind with our A. So this would be our creation of our message right here. So it would be ACU. GUA, which would have been created from our template, which would have been our DNA, which would have been TGACAT. Okay, so now we went ahead and created our mRNA piece. And what's going to happen here is we're going to go ahead and continuously make them. So again, notice that the mRNA is a complementary to the template strand. So again, it's a complementary piece or the opposites of our template right here. And it's nearly identical to the coding strand, meaning that this would have been the other side to it. So our mRNA is going to be almost exactly the same. Again, the only difference between this mRNA and this coding strand, or the opposite from our template strand, is, remember, DNA uses T versus mRNA is using U. So that's the only reason why it's not exactly the same. But other than that, you can see that they are incredibly close. We're using our template to create our mRNA, and our mRNA is very close to our coding strand. And then below is another example showing that you can see our template strand here, which would have been a complementary to our coding strand. But you can see that our RNA that's being created is, again, very similar. You can see all the letters are very similar. The only differences are those U's. All right, so now we went ahead and went through all that. We created our mRNA. We have that piece now. So after we're completely done, the mRNA is going to be processed in the nucleus and transported to the cytoplasm, where it is used in translation. So again, we went ahead and we have our DNA. It went ahead and opened up. We went ahead and copied it with our mRNA. So we're using the DNA as a template. We went through transcription process, make sure everything looks good. And now it's going to go ahead and leave the nucleus. So I can go ahead and go to the cytoplasm, where we're going to meet up with the ribosome, and we're going to start to utilize tRNA in order to transport our amino acids. We're going to chain those together in order to produce our polypeptide, which is going to be our protein. All right, so that is going to conclude our transcription portion of gene expression. So what I would like you to do is to go ahead and take a couple seconds, see if you can go ahead and answer questions 1 through 6, and then in the next slide, we'll go ahead and check it out. All right, welcome back. So let's go ahead and quickly look at some of these questions. So number 1, blank is the process cells used to copy DNA into a messenger RNA. So this is going to be transcription. Remember, you're transcribing or writing down a message because we're using our mRNA. Transcription occurs in the blank, so it's going to be the nucleus, and then it's going to go ahead and be transported to the cytoplasm. That's where translation is going to happen. So transcription is going to be the nucleus, and then translation is happening in the cytoplasm. What RNA bases would pair with the DNA sequence ATCGAC? So in this case, remember, we're looking for the complementary pieces. Now, if this was DNA to DNA, remember, we're using A and T, but in this case, since it's RNA, we're not using our T's, we're using our U's. So in this case, A goes with U, T goes with A, C goes with G, G goes with C, A goes with U, and then C goes with G. Right there, and you can see that would be the correct piece. What determines how the information in DNA is used to build a protein? It's going to be the order or the sequences of the nucleotide. Remember, our sugar phosphate backbone is important, but the bases itself and the order of that is going to dictate how and what proteins are being produced. All the following statements are correct except mRNA is a double-stranded molecule. That is incorrect. Remember, RNA is going to be a single strand, so it's going to look something like this, versus DNA are double-stranded, something like that. And then finally, RNA is built from which of the following? Select all correct answers. Remember, it's going to have a sugar, it's going to have a phosphate, and bases, and the bases are going to be A, C, G, and U. Now, although we are going to be using our mRNA to produce proteins, the mRNA itself does not contain proteins. It's the bases, the sugar, and the phosphate. All right, so let's go ahead and take a look at our second step when it comes to gene expression. In this case, it's going to be the translation. So when it comes to translation, just like the name suggests, we're translating that message now. So we went ahead and spent all that time. We used a template of DNA in order to produce our mRNA. The mRNA has now left the nucleus and it's in the cytoplasm, trying to find that ribosome in order to start making those proteins. So again, translation, we're going to be reading those instructions now. So the DNA/mRNA instructions are written in that nucleotide language, which is going to be our 4 bases. Remember, we had our A, C, G, and T for DNA. We use this as a template in order to produce our mRNA, where we were using uracil instead. So, however, protein language is written in amino acid language. So this is going to be something that's really important. And we're going to basically be trying to translate now, again, that nucleotide or those bases into our amino acids. And there's going to be a very specific and special way we're going to do so. So let's go ahead and take a look at our example, and we're going to look at how that's going to relate to the actual process when it comes to making our proteins. So remember, in transcription, we had our DNA, our master set of instructions found in the nucleus. We went ahead and made copies of that. Okay, again, the reason why we're making copies is because we do not want to lose our master instructions. Those are going to stay safe right in the nucleus. What's going to happen is we're going to make a bunch of copies though, because again, we need these in order to produce proteins. So now we made our mRNA. Now we're going to take these instructions, read them, figure out what they exactly mean in order to build our proteins, which are going to be our amino acids. Again, DNA here, you can see that that's going to unzip and open up. We're going to go ahead and make a complementary set from that. Our template set, which is going to, again, look very, very similar to our coding portion. And then we're going to use this, read this mRNA specifically in order to identify, because we're going to break them out into sets of 3, to identify exactly which amino acids are going to be needed in order to produce our protein. So translation is going to require 2 important molecules in order to translate the mRNA protein language. So the 2 we're going to need is our tRNA. tRNA is going to be known as transfer. Now this is a special type of RNA molecule. So each tRNA is linked to a very specific amino acid. Okay, so again, our tRNA is going to be linked to a very specific amino acid. The reason why that's so important is because our tRNA, we're going to go ahead and use our mRNA. Remember, we have that code, and what it's going to do, it's going to find the complementary piece. So if we had something like AUG, it's going to find the complementary set to this. So in this case, it would be U, A, and C. So our tRNA would have this codon, and it would find this here in order to know what amino acid it's going to drop off. Now, the ribosome is going to be another very important thing. This is going to provide a structure in which translation can take place. Okay, so again, you can see that we have our mRNA here. All of this over here is going to be our ribosome. And then our tRNA transfer, remember, is going to come in here in order to drop off our amino acids in order to produce our proteins. So again, our ribosome is giving us a structure and a place to do that translation. So when it comes to that tRNA, again, tRNA is very, very important. We talked about how it has something known as an anticodon. Again, our anticodon is going to be the opposite of our codon from our mRNA. So these are going to be incredibly important because remember, our tRNA is going to be responsible for a very specific amino acid. And we're trying to chain all of these amino acids together in order to produce a polypeptide. So again, the tRNA is going to be responsible for reading and translating the 3-letter codes, because again, we're going to be setting these in codons, so sets of 3. And then once it identifies where it's going to go, because again, they're going to fit in very specific spots. Once it's identified this here, again, our tRNA is going to come in, connect up to here, and it's going to say, hey, this is our complementary piece. So I know that amino acid 1 needs to be here. It's going to drop this off. And then our second tRNA is going to come in. It's going to look for a codon. It's going to find the part that it's going to be complementary to, and then it's going to say, awesome, this is where the second amino acid needs to go. So now they're chaining these 2 together, and then so on and so forth. Our third is going to do the same thing, find the place, chain it together. Fourth, do the exact same thing, find where it needs to go, its complementary set, and it's going to go and chain that together, and then we're going to start producing our protein chain. Okay, so this is going to be what it kind of looks like, but in a more kind of technical aspect. Again, you can see our mRNA is going to go this way, and it's going to start reading using our tRNA. And our tRNAs, you can see, have our codons, or in this case, our anticodons. It's going to find exactly where it needs to slot right into. You can see that this is going to be complementary right here to this. So it knows this is the position it needs to go into. It's going to go right here, go into the ribosome, and then it's going to go ahead and start linking all of these amino acids together in order to start producing our protein. And then what's going to happen is our tRNA is going to release after the amino acid is removed. So again, this is how we're carrying, or the tRNA is going to be transferring or transporting our amino acids, figuring out where it needs to go, connecting them together, and then it's going to go ahead and leave. Okay, so again, the mRNA is read one codon at a time, right? So one codon, which is sets of 3 bases, and the amino acid matching each codon is added to a growing protein chain by the ribosome. The mRNA is shifted one codon over in the ribosome, exposing a new codon for reading. The next tRNA moves in place or into place, and it's going to use that base pairing with the new codon. And then again, the cycle is going to continue until the protein chain is completed. So how do we know exactly what codons are going to code for what? So this right here is going to be known as our codon chart. And this is going to be incredibly important because you can see that we have all of these different sets or different pieces of codons, and you can see they're all coding for specific things. So this is going to be the dictionary of our codon amino acid relationship. And again, this is going to be called our universal genetic code because it lets the cells decode the mRNA into a chain of amino acids. The universal genetic code is the instruction manual for all cells, from bacteriums to humans, used to read DNA sequences from a gene and build corresponding proteins. So one codon, AUG, codes for the amino acid MET, and also known as our start codons. So one thing that's really important is in order for the body to know when to actually start producing proteins or grab our amino acids is we need to look out for AUG. AUG is going to be our start codon, again, letting us know this is where we need to start producing our protein. Now, it's going to go and start producing all the way up until it hits these specific ones right here, where it says stop. Now, what this is going to tell us, it's telling us, hey, we're done producing. We can stop now. Our protein is completed. So these are going to be important to remember. MET, which is AUG, is going to let us know when to start producing our proteins. And then we have 1, 2, 3 stop codons, UAA, UAG, and then UGA. And those are all going to let us know when to stop producing proteins. So the next thing I'm going to quickly talk about is how we're actually going to read these. So remember, we have sets of 3, which is going to give us a codon. So let's say we're going to go ahead and try to identify AUG. So you can see we have 1, 2, and 3 letters. So the way this works is we're going to start off with our first letter here. So we're going to go ahead and go to A. Our second letter, which is going to be a U over here. And then finally G, which is going to be over here. So the way we read this is we're going to go ahead and look at our first letter, A, and we're going to go ahead and move down here. We can see that our first one is a U, so this is not going to be relevant for us. So that means we can go ahead and cut or cross out all of this over here. The reason why is because this entire row right here does not have an A. None of these are starting with A, so we don't care about any of that. Then our second, C, same thing. None of that is going to be relevant for our AUG because, again, you can see every single one of these are going to start with a C. But going here, you can see that this entire row here is all going to start with A. So that means this is the only part we're caring about. So the G's, we do not care. So just like that, we went ahead and we have greatly reduced the amount of options that we need to look at. So we're only focusing on this portion right here now. In our second letter U, we're going to do the same thing, but we're looking at this top part. So we can see U right off the bat, meaning that we're only focusing at this part where they intersect now. Okay, so now we're only focusing on this interior portion here. Everything else we don't care about because, again, the second letters for all of these are going to be C, E, A, or G. So we do not need to worry about those. Finally, our last letter is G. So we're going to do the same thing. We're only focusing now on this specific portion. And then the way we're going to do this is we're going to read across this way. So you can see that our U's over here, every single codon is going to end with a U. But you can see ours ends with the G. So this row we don't care about. Again, next one's C. We're going to cross out all of those because that doesn't pertain to us. A doesn't match here, so we're going to cross all of those out. So you can see that our G is going to lead us to this, meaning that it's going to be MET. So that's going to be how you're going to read or use this chart in order to decode our mRNA. Remember, we're looking at our mRNA in order to figure out what amino acid needs to be placed there. Now, one thing that's important is you don't need to memorize this, but again, you should know how to use this. So this is going to be pretty important. So summary of transcription versus translation. Remember, we're starting with DNA. We're going to have our template right here. And what's going to happen is our mRNA is going to go ahead and try to make a copy. In this case, you can see our mRNA is going to find the complementary pieces to our DNA template. So you can see G goes with C, C goes with G, A goes with U. Remember, we are no longer using T's. DNA to DNA, we use T, but DNA to RNA, we're using our U's. So once we've done that, what we're going to do now is we're going to go and translate that. So we're going to go ahead and take a look at our codon chart, and we're going to look for a CGU. Once you find CGU, you can see that our protein is going to be ARG, or our amino acid is going to be ARG. So that's how we're going to go ahead and figure out what that's going to code for. We're going to do the same thing here. We're going to take our codon UCA. We're going to find what that is on our chart, and you can see it's going to be SER. Same thing, UGG. We can find out that that's going to be TRP. And then finally, ACU, giving us THR. And you can see we're chaining all of these amino acids together in order to make our protein. So now that we've identified exactly what that really means, one thing we need to look at is exactly how this is going to be read and what that's going to look like, and what happens when we have some mistakes. So the reason why we look at codons or sets of 3 is because it helps us really read and understand what those amino acids are. If we were to go ahead and take a look at this as an example sentence, you can see right here, because we don't have any of those pieces separated out, everything's going to be one big jumbled piece. But when we split them into our 3 individual words, now this one gigantic kind of mess is now readable and easily understandable. This you can't really even read, but once we split those out, you can see the sun was hot, but the old man did not get his hat. Again, we can easily understand and read this now. But when it comes to translation, sometimes there are some issues in transcription translation where we end up with words that shouldn't be there or things that get shifted. We either add an extra piece or we're removing a piece. Those are going to be known as our mutations. And this is how that's going to kind of completely change everything. If you were to shift this, you're shifting the reading frame. So now you're ending up with something that is completely different and completely unrecognizable. So instead of taking this, which is the code we actually want, if we shift it, you can see now everything is changed. And now, again, everything now is illegible. And then again, if we add something, so again, if you, for example, if we were to add a D for the old, or missing or added, you can see that it's going to, again, completely change everything. The sun was hot, but this doesn't make sense, and this doesn't make sense, this doesn't make sense, none of this makes sense now. So again, when we miss, remove, or add something, again, it's going to completely change everything. So if we shift the way we read it, it becomes illegible. If we add something, then again, or remove, you can see those parts are no longer legible anymore. And then if we misspell something, right, add something in on accident, it changes the entire frame of the sentence. So the sun was hot, but the old man did not get his cat. So now instead of talking about a hat, talking about a cat, which again is going to completely change everything. And the reason why I'm kind of talking about this is, again, it's going to lead us into something known as mutations. Once we have a mutation, there's essentially going to be a permanent change in the DNA sequence. So changing a gene sequence can change the amino acid sequence of the proteins it codes for. And mutations in DNA can lead to production of abnormal proteins and malfunctions that can cause diseases. So it's really important because, again, when we're going through this, and when we're trying to make these proteins, we want to make them exactly how the DNA is telling us to make them. But if we mess up through transcription translation, all of a sudden, we're going to have something completely different. Or if we have a mutation in our DNA, it's going to code for something completely different. Looking at this over here, you can see we have a normal DNA sequence, and this is what it's supposed to code for. You can see our DNA right here. We have everything perfect. A goes with T, C goes with G. Everything's exactly where we want it to be. Then we're going to use our DNA, remember, to make our message mRNA. You can see right here, we have that. That looks great. We're going to separate those out into threes, just like that. And then we use our codon chart in order to figure out exactly what amino acids we need. So MET right here, ASP right here, ALA right here, and then PHE right here. We're going to go ahead and make our proteins. Remember, proteins are going to fold on each other, and we have this piece. This is the shape we want it to look like. This is exactly what we need in order for it to do its job. Now, when we have our mutation, you can see that we have our CG. So instead of TA, TA, AT, AT, CG, 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 we have that extra piece. So because we've added this now, you can see our mRNA is completely different as well. You can see over here that our AUG is still the same, but our second piece, our second codon now, right here, instead of a GAU, now it's going to be GGU. So that means instead of our ASP that we want, we now have something completely different because we've placed an incorrect protein because our message was telling us to make this because they got it from our DNA and it had a mutation. So there was a change in that sequence. So now instead of MET, ASP, ALA, and PHE, we have MET, GLY, ALA, and PHE. So now again, we have this amino acid that is not supposed to be here. And now you can see it's going to create a completely different protein. So instead of this specific shape we want, in order for it to do its specific job, you can see now it's all big and bloated, and it's no longer going to really be able to do the job that it wanted to or needs to do, which again can cause a lot of issues. Again, production of those abnormal proteins can cause genetic disorders or diseases. So again, that's kind of what mutations are going to be. All right, and just like that, that is going to conclude this week's lecture. We covered a lot today. One of the things I highly suggest if you do have some trouble kind of understanding, or if you're a little confused, I highly suggest you definitely check out the supplementary material. Obviously, you're always more than welcome to reach out to me. But again, there was a lot of stuff, so please make sure you're taking time to review this lecture, and also make sure you're checking out Dr. Katz's lectures as well to get a different perspective when it comes to our transcription translation. So just as previously, what I want you to do is I want you to go ahead and pause this, see if you can answer these following questions, and then we'll go ahead and check them out together. All right, so coming back, this is what you should have gotten. So translation requires a transfer and tRNA to decode and build a protein. What tRNA anticodon would match with the mRNA codon AUG? Remember, we're just figuring out what the complementary set's going to be. When it comes to RNA, remember, it's going to be A, U, then C, and G. So over here, you can see A should go with RU, and then our U should go with A, and then our G should go with C. You can see that is exactly what that is right there. Using the universal genetic code to translate the mRNA sequence, so we have two codons here, ACC and then GUC. Remember, what we're going to do is we're going to go ahead and figure out our first, second, and third letter. We're going to use that chart in order to place them, and then wherever they all kind of intersect is going to be our amino acid. So in this case, for these two, it should have been THR and VAL. Number 4, the codons for the amino acid proline includes all the following except which ones? It should have been CGC. Which codons tell the ribosome to stop translation? Remember, we have 3 stop codons, and those are going to be UAA, UAG, and then UGA. Finally, what would happen if the DNA sequence of a gene is altered? A mutation protein can be produced that malfunctions and causes diseases. All right, and that again is going to conclude our lecture. So as always, if you do have any questions, comments, concerns, please feel free to reach out, and I look forward to seeing everybody in our next lecture.